Welcome to Anatomy and Physiology class, the best channel to explore the wonders of human body. If this is your first time to visit this channel, like, subscribe, turn on notification and follow all our social media accounts to get more updates on our programs.
Our topic of today is cell. Remember that this tutorial is for this category of students, so don't expect broad explanation from us. So this tutorial is for students in diploma programs, so don't expect broad explanation from us. And this is the content that we are going to discuss concerning the cell. So what is a cell? So cell is the structural and functional unit of an organism, or is the basic unit of an organism. When we say structural and functional unit, means it is the cradle of the body structure and its functions. It is the smallest unit, or it is the smallest point where all the body structures started and all the body functions started, because all the body organs are made up of cells, and all the functions that are carried out by the body parts or organs, they started at cellular level before they manifest at higher level where they can be seen with naked eyes. So component of cell, what does the cell compose? So the cell composes of, it has two major components. It has the cell membrane, which is the outer covering of the cell, and the cell cytoplasm, which is the internal environment of the cell. So this covering, it is what is representing the cell membrane here, and this internal part of the cell, where it contains so many cellular organelles. This is the cytoplasm. So cell membrane. So cell membrane, it is the outer covering of the cell, and the cell membrane is a double-layered membrane, hence called the lipid bilayer. So we'll explain what lipid bilayer means. So the cell membrane, it has so many chemical components that are present on it. It is made of macromolecules, which include lipids, protein, and carbohydrates. So starting from the lipid component of the cell, each and every lipid molecule of the cell, it composed of a head and a tail. The head is made of phospholipids, combination of phosphorus and lipids, and it is hydrophilic in nature, which means it allows water soluble substances to pass through it. Then the tail is composed of fatty acids, and it is hydrophobic. It composed of fatty acid chain, and it is hydrophobic, which means it doesn't allow water soluble substances to pass through it, which means so whenever there is any water soluble substance, even if these hydrophilic functions allow it to become mixable with it, so this hydrophobic function will not allow it to cross the cell membrane. So the hydrophobic function is only allow lipid soluble substances to pass through it. That is why you see these yellowish substances are cholesterol. You know that cholesterol are fat soluble, so they can pass through this lipid bilayer. So that is that about the lipid component of the cell. Then the protein component of the cell. The protein component of the cell, there are different combination of proteins that are present within the cell membrane, and they are categorized into two. We have the intrinsic or integral proteins and the peripheral or extrinsic proteins. The intrinsic or integral proteins, they are also known as transmembrane proteins. They are proteins that pierces the entire thickness of the cell membrane. It crosses the entire thickness of the cell membrane, thereby allowing some substances to pass through it. Some of these integral proteins, some of them are channel proteins. Channel proteins are proteins that allow water soluble substances to pass through them. For example, this one. And some of them are enzymes. Some of them are carrier proteins. Some of them are antigens, and some of them are receptors for some hormones. Then the peripheral proteins. Peripheral proteins are proteins that doesn't cross the entire thickness of the cell membrane. For example, this protein. So you can see this protein, it is just present on only one part of the cell membrane, either internal or external part of the cell membrane. So also this one is an example of peripheral or extrinsic protein. So some of the example of the extrinsic protein include, some of them act as carrier protein, some of them are enzymes. Then the carbohydrate component of the cell membrane. So the carbohydrate component of the cell membrane, the carbohydrate is always in contact with either protein or lipid. Those that are in contact with protein, they are called glycoprotein, while those that are in contact with lipid, they are known as glycolipids. So the carbohydrate contains long chain of different kind of carbohydrate subunits. So that is why it is in form of polysaccharide that is in contact with either protein as in glycoprotein or lipid as in glycolipid. And some of these carbohydrates also act as enzymes and receptors. So what are the functions of the cell membrane? So the cell membrane separates the ICF from the ECF. ICF means intracellular fluid. There is fluid inside the cell, which is known as cytosol. So the fluid inside the cell membrane is known as cytosol. So this cell membrane it separates this ICF, the fluid inside the cell, intracellular fluid. It separates it from ECF, extracellular fluid, the fluid outside the cell. Then protection. So another function of the cell membrane is to protect the cell. It protects the cell from any invading substance, any harmful substance that will try to enter the cell. The cell membrane will now protect it. It will not allow that harmful substance to get into the cell. Then selective permeability, which is one of the most important functions of the cell membrane that composed of different subfunctions, and we will discuss this when we come to transport across cell membrane. So selective permeability means it selects some substances to pass through it, and it doesn't allow some substances to pass through it, which is very, very important. Absorption. So the cell membrane is responsible for absorption of various important substances that are used by the cell, which include the respiratory gases like the oxygen, carbon dioxide, nutrients like carbohydrate and protein, hormones, etc. Excretion. So harmful waste substances or metabolic waste that are produced as a result of cellular activities, they are excreted by the cell membrane by different mode of transport across cell membrane, which we are going to see later. And shaping the cell. The cell membrane is responsible for providing the shape of the cell. Even though the cell membrane it is not the only part of the cell or only component of the cell responsible for this, there is some contribution of cytoskeleton, which we are going to see what cytoskeleton means later. So the cell membrane is responsible for giving rise to the shape of the cell. Then cell division. So immediately after the genetic makeup of the cell is divided into two, and the cytoplasm of the cell is also divided into two, and the cell membrane is the one that is responsible for dividing each and every mother cell or parent cell, dividing it into two daughter cells. So the cell membrane it plays an important role in cell division, which we are also going to see that later. And the cytoplasm of the cell. So the cytoplasm is an internal part of the cell. It contains of a fluid known as cytosol. So this cytoplasm it contains water. It contains a very large percentage of water and a fluid known as cytosol. Then cell organelles. So it contains fluids and organelles. That is what is present inside in the cytoplasm of the cell. The fluid is combination of water and this cytosol. Then the cell organelles, which we are going to see each and every one now. So let us start with nucleus. So nucleus is a spherical organelle. It is bounded by a double membrane. And it has so many pores on its membrane, which are known as the nuclear pores or holes. It has so many holes or pores on its membrane, which are known as the nuclear pores. And the nuclear membrane is also known as the nuclear envelope, as you are seeing it here. So inside the nucleus, it has a peripheral part which is known as the chromatin, and it has a central part which composed of the nucleolus. The chromatin composed of uncoiled DNA. Then the nucleolus composed of RNA. So what are the functions of the nucleus? Nucleus controls other organs. So the nucleus is responsible for the control of the cellular activities in the cell. It is the one that is responsible for controlling the remaining organs, hence known as the brain of the cell. And synthesis of RNA. The nucleus is responsible for synthesizing RNA. We are going to see that when we come to protein synthesis. Then storage of DNA. The genetic makeup of the cell is stored inside the nucleus, here in the chromatin, which is the peripheral part of the nucleus. Then cell division. So the nucleus is responsible for dividing the genetic makeup of the cell into two equal halves, thereby giving rise to the daughter cells. Then endoplasmic reticulum. Endoplasmic reticulum is a tubular organelle which is also bounded by a double membrane. And this endoplasmic reticulum is divided it is of two types. We have the smooth ER and the rough ER. So the smooth ER is the endoplasmic reticulum that doesn't have ribosomes that are bound to it, while the rough ER is an endoplasmic reticulum that has so many ribosomes that are bounded to it. So this is the structural difference between the two. Then the functional difference between them is that in general, in the endoplasmic reticulum, the rough endoplasmic reticulum is responsible for synthesis of protein. This reddish one, the one in the red color, is the rough ER. So it is responsible for synthesis of protein and proteinous substances like peptide hormones, example insulin. So the rough ER is responsible for production of it. Then degradation of one-out organelle. An organelle that is a useless organelle, or organelle that is not capable of performing its functions. So that organelle is destroyed by rough ER. Then the smooth ER. The functions of the smooth ER include synthesis of lipid and steroid substances like steroid hormone. Example of steroid hormone include the estrogens, progesterone, and other sex hormones are example of steroid hormones. Lipid like cholesterol. So all these substances they can be synthesized by the smooth endoplasmic reticulum. And detoxification means removal of harmful toxins. Then Golgi apparatus. Golgi apparatus is also a tubular organelle. It is also a tubular organelle, only that the tubules of Golgi apparatus they are parallel to one another. They are not. It is not continuous or as to the adjacent tubules, just like that of the endoplasmic reticulum. Rather, the tubules of the Golgi apparatus or Golgi bodies, they are parallel to each other. They are not connected. So So Golgi apparatus is also bounded by a double membrane. This double membrane, so the membrane that bounds or that covers the cell organelles is also known as plasma membrane. So functions of Golgi apparatus, it includes packaging and processing of materials. So for example, after the proteinous substances are produced by smooth ER, or the lipid substances are produced after they are produced by the smooth ER, or after the proteinous substances are produced by the ER, they will be sent to Golgi apparatus. Now Golgi apparatus, it will now package, it will now process them. If they need any modification, it will now modify them. That is the processing. Then packaging, it will now put them inside secretory vesicles. Look, these are the secretory vesicles that the Golgi apparatus package substances in them. So processing and packaging of materials. Then another function is labeling and delivery of materials. Now each and every vesicle, it will now label each and every vesicle. For example, to say this vesicle contains proteins, this vesicle contains lipids, this vesicle contains hormone, this vesicle contains enzymes. It will label each and every vesicle to an extent that whenever it sends a vesicle to specific organelle or to specific site of the cell, that site or that organelle will now be able to identify that vesicle. It will now be able to differentiate that vesicle from other vesicle. That is by the labeling. Then delivery of material. Then it will now deliver or send those vesicles into their appropriate site where they are supposed to be sent. That is why the Golgi apparatus is known as the post office of the cell, and it is also known as the shipping department of the cell. And lysosomes. So lysosomes have the thickest membrane. So they are also bounded by a double membrane, and their membrane is the thickest membrane because they contain hydrolytic enzymes that are capable of destroying the cell, or that are capable of destroying so many organelles or so many structures that are present within the cell. So that is why lysosome is bounded by a double membrane. The functions of it is bounded by a double membrane and it has the thickest membrane. So the functions of lysosomes include degradation of macromolecules. So macromolecules that are in complex structures. For example, if a carbohydrate is in form of disaccharide or polysaccharides within the cell, so lysosome is the one that is responsible for degrading it or further digesting it into smaller subunits. Then degradation of worn-out organelle. So whenever there is a worn-out organelle, lysosome is also responsible for degrading or destroying that worn-out organelle. So what we are trying to say here, previously we had said that degradation of worn-out organelle is a function of rough endoplasmic reticulum. So also here we are also saying that the degradation of worn-out organelle is also a function of lysosomes. What happened is that the rough endoplasmic reticulum is responsible for wrapping and putting that worn-out organelle into a vesicle. Now after that vesicle is known as autophagosome. Then that autophagosome will now bound to a primary lysosome. A primary lysosome is a lysosome that contains unactivated enzymes. So that primary lysosome will now bound to that autophagosome. So when it's bound to that autophagosome, it will now become a single vesicle, and those unactivated enzymes will now be activated so that they will now destroy the worn-out organelle. So the function of degrading of worn-out organelle is for both lysosome and the rough endoplasmic reticulum. So when the lysosomes bound to that, when it's bound to autophagosome, where it becomes a single vesicle, it is known as secondary lysosomes, where the enzymes that are present inside it will now become activated and they will now destroy the worn-out organelle. Then peroxisomes. Peroxisomes are also bounded to a double membrane, and the functions of peroxisomes include breakdown of fatty acids. So fatty acids, they are they are broken down, or they are further digested by the peroxisomes. So also the detoxification. Detoxification is also a function of peroxisomes, where it detoxifies or where it removes the harmful toxins, or it destroys the harmful toxins that are present within the cell. Example like alcohol, drugs, and other toxic substances that are no more useful to the cell. So the peroxisome is one responsible for destroying them or removing them. So peroxisomes contain oxidative enzymes that are mainly ready for that function. Then mitochondria. Mitochondria is an oval organ. It is also bounded by a double membrane. And mitochondria, it is the only organelle that contains DNA apart from nucleus. So apart from nucleus, mitochondria is the only organelle that contains DNA. And that DNA is known as the mitochondrial DNA, while that of nucleus is known as the nuclear DNA. So the functions of mitochondria. So the function of mitochondria is the powerhouse of the cell. It is the one that is responsible for generating the electricity or the power that will be used in the process of cellular activities. So also another very important function of mitochondria is apoptosis. Apoptosis is a programmed cell death whereby the cell destroy itself, whereby the cellular component they get destroyed by all the cellular component they get destroyed by the cell without damaging the cell membrane. So apoptosis is a programmed cell death, and it is also known as cell suicide, when a cell is not capable of functioning very well. So apoptosis will now be activated, and that apoptosis degeneration. So the activation of apoptosis is under the control of mitochondria, and it plays a very important role in that cell suicide or programmed cell death. And ribosomes. So ribosomes are the smallest organelles, and they are not bounded by a double membrane. And there are of two types. Some of them are bounded to rough endoplasmic reticle, hence known as the bound ribosomes. Bound ribosome because they bound to rough endoplasmic reticle. While those that are moving freely in the cytoplasm, they are known as free ribosomes. So synthesis of protein is the main function of ribosomes, where we are going to see that when we come to protein synthesis. So ribosomes are also known as the protein factories. And centrosomes. Centrosomes are bounded by a double membrane. There are also organelles that are bounded by a double membrane, and it contains some tubular structures that are responsible for dividing chromosomes into two. It contains some tubular structures that are responsible for dividing chromosomes or chromatids during the process of cell divisions, and those tubular structures are known as centrioles. So the centrioles contains mitotic spindles, which are responsible for that function, where we are going to see that when we come to cell division. So their main function is in this process of cell division. And cytoskeleton. The word cyto means cell, and skeletal means framework. So the function, cytoskeleton is a combination of different proteins or cell organelles or fibrous cell organelles that are responsible for providing the shape of the cell, or for maintaining the actual shape of the cell. So the main function is they determine the shape of the cell and they give support to the cell, because without this cytoskeleton the cell will definitely collapse. So the difference between somatic and germ cells. What are somatic cells and what are germ cells? So somatic cells are the body cells that are not involved in reproduction. They are the cells that are not involved in the process of reproduction, and they are which means somatic cells are just the normal body cells that are not reproductive cells. And their examples include myocytes, which are muscle cells. Here is a myocyte. Nerve cell, which are neurons. Here is a neuron. Epithelial cell example, this is an epithelial cell. These cells are epithelial cells. Osteocytes are bone cells. They are not here. Adipocytes are fat cells, etc. So the example is there are many more. And germ cells. Germ cells are reproductive cells. Cells that are involved in the process of reproduction. An example include sperm cell and egg. So here is a sperm cell which is from the male side and egg from the female side. So these are the only germ cells. So this differentiation is very important when we come to the process of cell division. So the difference between some of the biomolecules that are present inside the cell, the nucleic acid that is present within the cell. So it is of two types, DNA and RNA. So actually this topic, difference between DNA, RNA and chromosome, it is not actually part of the curriculum. We just inserted it. We inserted it just to help you to understand the process of protein synthesis and cell division. Because without understanding the actual structure of DNA, RNA, and chromosomes, without knowing the differences between them, will not actually comprehend what protein synthesis or cell division is all about. So RNA is a nucleic acid that has a single strand and that is made up of single collection of nucleotides. Here are the nucleotides that are present on RNA. They include adenine, guanine, cytosine, and uracil. Adenine, guanine, cytosine, and uracil. These are the nucleotides that are present in RNA. RNA is of different types. We have the messenger RNA. The messenger RNA, it is synthesized inside the nucleus. Then it is sent out of the nucleus, where it passes through the nuclear pores to enter the cytoplasm. Then we have the transfer RNA. Transfer RNA, it has a very important role in arranging or in bringing amino acids, thereby dropping them to form a long chain of amino acids in the process of protein synthesis. Then ribosomal RNA. Ribosomal RNA is found inside ribosome, which is the one that is responsible for arranging different amino acids, thereby forming a complex 3D shape of protein. So ribosomes can be found inside the nucleus, in the nucleotides, then in the cytoplasm, as in transfer RNA and some messenger RNA. Then DNA. DNA stands for deoxyribonucleic acid. unlike RNA, which is ribonucleic acid. So RNA composed of a ribose sugar. I don't want to go deep into the biochemical structure of the RNA so that you will not get confused. So DNA composed of a deoxyribose sugar. DNA stands for deoxyribonucleic acid. It has some base pairs on it. The shape of DNA is a double helical structure. It's a double helical structure, unlike RNA, which is a single helical structure. So DNA is a double. It has a double helical structure, and each and every segment of DNA from one segment to another segment is known as gene. And the structure of DNA is formed by these base pairs, which include adenine, which pairs to thymine, then guanine, which pairs to cytosine. So here in the structure of DNA, we are not having uracil. We are having thymine instead of uracil. So these are the base pairs that are present within the structure of DNA, and they are the one that form the genetic code of each and every organism. So DNA is found inside. DNA can be found inside the nucleus. So also it can also be found within the mitochondria, as I said earlier, where we are having the mitochondrial DNA. So what is a chromosome? So chromosome is a coiled DNA. Chromosome is formed if DNA coils itself on a protein known as histones. So it forms a complex structure known as chromosome. This is a structure of chromosomes. These arms of chromosomes are known as the telomere. So chromosome, it has a short arm and long arm. This long one is the long arm, and it has a short arm. In the center where the two chromosomes meet together is known as the centromere. So this one is a chromosome, and this one is also a chromosome. So this is a pair of a chromosome. This is a single pair, or this is a pair of chromosomes, which compose of two chromatids. And cell division. So cell division is the process by which cell divides to form a daughter cell. So remember that each and every cell, in the humans, there are about 23 pairs of chromosomes, making 46. So out of those 23 pairs, 22 are chromosomes, and only one pair is X chromosomes, where we are having XY for males and XX for females. So the types of cell divisions, the two main types of cell divisions are mitosis and meiosis. So mitosis is a type of cell division whereby a cell divides itself to produce two daughter cells with diploid number of chromosomes. Diploid means the complete number of chromosomes, means twice twice the half number of chromosomes, means 23. That is diploid chromosomes, making 46. So when we say diploid, it means 23 pair of chromosomes. So a cell that is having 23 pair of chromosomes, it is known as diploid cell. So you can say mitosis is a type of cell division that occurs in somatic cells, because mitosis took place in the normal body cells, unlike the germ cells, sperm and egg, where meiosis takes place. And mitosis, you can also say mitosis is a type of cell division whereby it's a type of cell division that occurs in diploid cells. So meiosis is a type of cell division whereby a cell divides to form haploid chromosomes, to form four daughter cells, or to form cells with haploid chromosome number. Or you can say mitosis is a type of cell division that occurs in germ cells, and it requires two rounds of cell division. So mitosis requires two rounds of cell division, why we are going to see that later. Now let us start with mitosis. So the steps of mitosis include prophase, metaphase, anaphase, and telophase. So in prophase, you have to take note that prior to cell division, the DNA of the cell, it replicates itself to produce twice the normal amount of DNA. The cell replicates its DNA to produce twice the normal amount of DNA it is having, which means that DNA that is originally capable of producing 23 pairs of chromosomes, now it will be replicated so as to produce the DNA that is capable of producing 46 pairs, not 23 pairs of chromosomes before cell division, that is at a space of interphase. So now in prophase, the nucleolus will disappear and DNA will now coils to form chromosomes. Then the nuclear membrane will also disappear and centrosome will now appear. That is what happened in prophase. Then in metaphase, the chromosomes will now align themselves at the center, and those centrosomes with centrioles and mitotic spindles will now they will now start to move toward the center of the cell. They will now start to move toward each and every chromosomes, where they will now attach to the chromosomes at the centromere. Then in anaphase, those mitotic spindles or kinetochore fibers will now divide each and every chromatids. It will now divide each and every chromosomes into two sister chromatids, and now each and every group of sister chromatids will now be aggregated at the two ends of the cell. Then telophase. In telophase, now there are two collections of chromosomes. The chromosomes will now uncoil to become DNA. Nucleolus will appear, nuclear membrane will also appear, and those mitotic spindles, they will now disappear. Then the cell will now start to divide its cytoplasm, and the cell membrane will now start to divide the cell into two daughter cells. And this is the role of cell membrane in the process of cell division. So the division of cell into two daughter cells is what is known as cytokinesis. Cyto means cell and kinesis means movement. And meiosis. So as we said, meiosis is a type of cell division that occurs in germ cells, or is a type of cell division whereby a cell divides, produce cells with haploid number of chromosomes, and it requires two rounds of cell division. I remember that prior to each and every cell division, at the space of interphase, the cell replicates its DNA to produce a normal amount of DNA, to produce DNA that is twice its normal amount of DNA, which is capable of producing 46 pairs of chromosomes. So take note of this, because in this mitosis, after the cell is divided into two daughter cells. Each and every cell is having haploid, is having a diploid number of chromosomes. That means each and every cell is having 46 pair of it is having 46 chromosomes. That is 23 pairs, because that 46 pair, it is the one that will be divided into two equal halves, so as to produce two daughter cells that are having 46 chromosomes. That is 23 pairs. Now back to meiosis. So in meiosis, so the steps of meiosis include two major rounds of cell division. The first one is meiosis 1, or phase mitotic division. So in phase mitotic division it is having some stages also that are similar to that of mitosis: prophase, metaphase, anaphase, and telophase. So in prophase, the nuclear membrane disappear, the DNA of the cell, it coils to form chromosome. It coils to form chromosome, and the nuclear membrane also disappear. Then centrosomes with mitotic spindles appear. That is what happened in prophase 1. Then in metaphase, the chromosomes, they align themselves. Each and every chromosomes, they align themselves and share genetic material, and they share genetic and the homologous chromosomes, the chromosomes that are similar to one another, they share chromosomes, because the chromosomes are combined together. That is why there is any possibility of a chromosome to pick a genetic information of its sister that is close to it. Then in anaphase, the sister chromatids are divided into two, where we will have two groups of chromosomes in the where we will have two groups of chromosomes in these cells. Then in telophase, or telophase 1, the chromosomes, the two groups of chromosomes, now the nucleotides will now appear, and they will be encircled or enveloped by the nuclear envelope, which will now appear. Then those mitotic spindle will now disappear, and cytokinesis will now start to take place. So one of the difference between the meiosis and mitosis is the independent assortment, whereby the cells, whereby the chromosomes, they share their genetic material, which occurs in metaphase 1. Now meiosis 2. In meiosis 2, the stages are prophase 2, metaphase 2, anaphase 2, and telophase 2. So in prophase 2, similar to what happened in prophase 1, of course, DNA will now coils to become chromosomes, but here the DNA is not replicated. There is no any replication in the DNA. Previously, immediately after the meiosis 1, the cells that are produced, they are diploid cells there are two daughter cells with diploid number of chromosomes. Each one of the two cells is having 46 chromosomes, or 23 pairs. So now those cells that are produced, the daughter cells, they will now be further divided, where their chromosome number will now be divided into two, so as to produce 4 cells with haploid number of chromosomes. Now progress 2. In progress 2, the DNA coils to form chromosomes. Nuclear envelope and nucleolus disappear. Then centrosome with mitotic spindles appear. Then in metaphase 2, the cells align themselves at the center where the mitotic spindles attaches to each and every pair of chromosome at the centromere. Then in anaphase 2, the sister chromatids are divided into 2 and they are separated into 2 equal halves where they are collected at the 2 sides of the two poles of the cell. Then the two daughter cells will now be divided into 2 in telophase 2, where the nucleolus and the nuclear membrane will now appear. Then the mitotic spindles will now disappear, and that is what happened in meiosis 2, where 4 cells with haploid number of chromosomes are produced. So in females, we will be having 22 chromosomes plus X chromosome in each and every cell, while in males, we will be having 2 cells that are having 22 chromosomes with X. We'll having 2 cells that are having 22 chromosomes with X chromosome and 2 cells that are having 22 chromosomes with Y chromosome. So this is just a simple summary of what happens in meiosis, where the primary or parent cell is divided into two cells with diploid number of chromosomes. Then the 2 cells are further divided into each one is divided into 2 cells with haploid number of chromosomes. So 4 cells are produced with haploid number of chromosomes in meiosis. So this is meiosis 1 and this is meiosis 2. So there is something. So this is what actually happened. Protein synthesis. So protein synthesis is the process of making protein from genetic information, and the steps include transcription or DNA transcription and RNA translation. So transcription is the copying of segment of DNA into messenger RNA, while translation is the decoding of messenger RNA or transformation of the information that is present within the messenger RNA into sequence of amino acids, which are the building blocks or the subunits of protein. So this diagram explains what the protein synthesis is all about. So this diagram is just trying to represent the internal part of the cell, where we are having the nucleus, and the nucleus which is also present within the cytoplasm, and the free space of the cytoplasm that is out of the nucleus. So here is inside the nucleus. All this event that is present here is inside the nucleus, while here is just inside the cytoplasm, not inside the nucleus, even though the nucleus too is also inside the cytoplasm. So we said the steps include DNA transcription and RNA translation. So DNA transcription is the copying of segment of DNA into messenger RNA. What happens is that there is an enzyme that is known as RNA polymerase, which attaches itself to the terminal end of DNA, thereby dividing that double helical structure of DNA into two strands. So after it divides DNA into two strands, now the codes of RNA, now the base pairs of RNA will now come and copy that segment of DNA where we are. Remember we said where we are having adenine. Adenine normally pairs with thymine in DNA. So adenine will now pair to uracil in RNA. Then guanine will now pair to cytosine, and that is how the pair, that is how the coding continues until when a messenger RNA is produced. Now that messenger RNA will now be sent out of, it is the one that contains the code that contains the information of the normal protein that will be produced. So now that messenger RNA will be sent out of the nucleus via the nuclear pore, and after that ribosome will now attach to that ribosome will now attach itself to the terminal end of that messenger RNA, and after that then transfer RNA will now be carrying different subunits of proteins, which are the amino acids or building blocks of protein, which are the amino acids, and it will now be reading the code of messenger RNA. That is the decoding of messenger RNA into segment up into protein. So it will now be reading the code in the that is present on the messenger RNA, thereby inserting the corresponding amino acid that correspond to that code. So this one is these are the amino acids. This one is methionine, and this one is leucine, this one is glycine, this one is serine. Different kind of amino acid. When it reads the codes, then it will now the transfer RNA that correspond to this code will now attach to it. When it attaches and read the code, it will now release an amino acid, and when it releases an amino acid, then a long chain of amino acid will now be produced. So after that, then ribosomal RNA, which is not present here, will now go and attach to that long chain and transform that long chain into a 3D complex shape of protein. So that is what happens in the process of protein synthesis. Then mutation. So remember the shape or the segment of DNA that we shown you. So there are some abnormalities or conditions whereby that coding will now be altered or tempered. It will now be altered, or it will now be destroyed, or DNA will be produced whereby some segments of DNA will now be loose, or some codes of DNA will now become lost. So that is what is known as genetic mutation. So genetic mutation refers to the change in DNA sequence within a gene or chromosome of an organism, which result in creation of new character. So mutation is inheritable. So these segments of DNA that you are seeing, it is what is known as gene, and each and every gene it's encoded for a specific trait. Each and every gene is responsible for producing a specific trait in the human. Trait means character. For example, we have the gene for tallness, we have the gene for shortness, we have the gene for eye color, skin color, and different characters. So if the coding of that gene is altered, then definitely the trait or the character that will be produced from that gene will now be changed from normal to abnormal, because the code is tempered, and that is what is known as inheritable. And there are so many factors that are associated to mutation. When the mutation is spontaneous, it can be spontaneous, it's mostly spontaneous. It can occur just naturally, but there are some environmental factors that can lead to it, like the X-ray, ultraviolet rays, heavy chemicals like the lead. Some of harmful drugs can also lead to genetic mutation, or some drugs that are normal in mothers but abnormal to the embryo. Some drugs can also produce some genetic mutations, thereby producing some abnormalities in an embryo. So any factor that is responsible for producing a mutation is known as mutagen. And normally mutated cells they are self-destroyed. They are destroyed by, so the immune cells normally activate the process of apoptosis in the mutated cells, while some of them are destroyed by the immune cells itself. So apoptosis is activated in the mutated cells if the mutation is very severe. And take note that mutation, when it occurs in autosomes and when it occurs in somatic cells, it's not inheritable. But when the mutation occurs in germ cells, because germ cells are involved in process of reproduction, so that mutation it will now become inheritable. So that is that about this tutorial. Here are our reference. So thank you very much for having your time and watching this tutorial.